everyone. Welcome back to Science with Spice Between Experiments. I'm your host, Dr. Danielle Spice, science communicator and scientist. On Science with Spice Between Experiments, we talk to real-life scientists about their science, but we challenge them to talk about it in a way that is accessible to everyday people. In this episode, I'm super excited to introduce you to Dr. Pablo Andreas Munoz Llancao, who is a neurobiologist from Yale School of Medicine. And we're going to talk about everything that has to do with lysosomes and neurodevelopment. Lysosomes are these really cool organelles inside of our cells. So if you go, really inside of the cell, that are typically involved in removing basically garbage from our cells that we don't really need anymore. And Pablo tells us that maybe lysosomes actually do a lot more. And not only that, they're super important during development of the brain, and that there are these newly discovered mutations that exist that cause neurodevelopmental disorders because of the lysosome. So we're going to get all into that, and I'm super excited for us to talk about it. Hi, everyone. I am so excited today to be talking to Dr. Pablo Andreas Munoz Llancao, who is a neurobiologist and an associate research scientist in the Department of Cell Biology and Neuroscience at Yale School of Medicine. His work focuses on how disruption of lysosome transport in neurons can lead to neurological disorders using both animal models and stem cell-derived neurons to understand these different mechanisms during brain development. He's originally from Chile, and he completed his undergraduate studies at the Pontifical Catholic University of Chile and actually has a double PhD between Chile and the Netherlands through the Abel Tasman Talent Program at the University of Groningen and the University of Chile, where he investigated the role of different signaling pathways in axon determination, which is a part of our neuron. And in previous positions, he studied how different signaling pathways like cyclic AMP and AKT signaling contribute to sex differences in brain function in mouse models of intellectual disability and autism. And more broadly, how intracellular signaling and cytoskeletal regulation shape neuronal differentiation and polarity. I know there's a ton of science words in that, but we're going to get into it. And Pablo, I'm so excited to talk to you today. How are you?
Hi, thank you, Daniel, for the invitation. It was great to meet you.
So now you're currently an associate research scientist and you were a postdoc before. And I feel like if you're not really well versed in the language of academic appointments and positions, being an associate research scientist, like, what does that mean? Can you tell us what your job is?
Yeah. Yeah, so as a PhD student, you are basically learning how to do research, how to be a scientist, and you need a constant supervision and trying to guide you. My problem as a PhD student, I was like, sometimes I was very I lost my focus. No, I was just going to a rabbit hole and some, experiment, and I couldn't push forward some part of the project. So my supervisor was there to keep me like in a straight line and everything. So as a postdoc, you are more independent, and you take part of a project in a lab. And since you are more independent, you can have more liberty to think about possible experiment to do without your supervisor, and then you discuss that. And associate research is basically just like a senior postdoc. You are very independent. In my case, I drive my own my project and my way, with the discussion of the PI of the lab. And we both agree to do that and to drive the research forward. But basically, yeah, an associate researcher is still like a senior postdoc, but under other, on another PI lab. No, you still know you don't have your own PI, so you are building your career for that. Yeah.
I was gonna say, it sounds like you're the best in the lab than you could ever possibly be realistically, because you're the most senior of the postdocs, but you don't have the added pressure of having to constantly be writing grants to make sure the lab is well funded. Unless, I'm assuming as an associate researcher, you probably have to write some grants.
Yeah. Yeah, I had to run my grants, but if But if I don't get it, still I have the support
Yes.
of the PI, no?
Yes.
So still, yeah, the grant I try to apply, they can help me for some tools, but
Mhm.
still you are under umbrella of the resources of the main PI of the lab, which is Sean Ferguson. Yeah,
Yes.
yeah.
Okay. So, talking of being in Dr. Sean Ferguson's lab at Yale, can you really break down what is your current research project in his lab as if I don't have a PhD? I'm just a regular person on the street. And can you tell us why is the lysosome so important for the brain?
So I just returned this week from a conference in lysosome where
Great.
you can take a little bit your perspective because from our high school or the basic undergrad books, they always define the lysosome as the garbage truck inside the cell. They eat
Hmm.
the garbage. And now recent research, and we know that the lysosome, which is a organelle inside every cell, like a little compartment, beside to degrade things that the cell don't need, they're actually are half of signaling. They can sense some nutrients, send dysregulation and metabolism, and send signaling and contact other compartment inside the cell. So they have more function than just degrade stuff, you know? And so why it's important is this regulation of the lysosome being associated with neurodegenerative disease like Alzheimer, Parkinson, ALS. Um, what they have in common is because this disorder sometimes accumulate some protein there, they can affect the neurons. And since the neuron cannot divide, they cannot not dilute the garbage inside, so they start to accumulate dangerous protein. They aggregate and create some cell death. So the lysosome cannot hold and maintain properly the health of the neurons in particular.
Hmm.
So they start to suffer. And not only the neurons, there are other cells in the brain like microglia, astrocyte, they also can be affected if the lysosome is not functioning well. So the laboratory of John is studying the lysosome in different ways, from the stress of lysosome in macrophage cells, and also in particular with a focus in Parkinson disease, and also the basic how lysosome transport inside the cell, in my case, inside the neurons, or how other proteins regulate the transport of lysosome, how they get affected by environmental insults or stress or anything. And in my case, Sean had a mouse model that needed to be described. And this model is based, it is a mice, they have a human mutation. They cause a rare neurodevelopmental disorder. So in 2019, two papers came out telling them they found in a group of people in Japan, and they have a particular and severe neurodevelopmental disorder that it causes a global developmental delay and affects severe the brain, the nervous system. The kids have a range between intellectual disability, autism. Some of them present seizures, but they are characterized to have severe motor problems. So we have that animal. So I was hired to okay, we have this model. We need to know what happened. There's nothing that we know about this.
Hmm.
There was some kind of hint in those papers that what this type of mutation can do in the neurons, and shown years before I arrived to the lab, they published what happened in the neurons when you remove GIP3 from the neurons, because GIP3 is the protein that I am studying. It is highly enriched in neurons. If you remove it, the neurons have swelling in the axon. They start to accumulate lysosome, but there's no disease associated with the lack of GIP3 because it's lethal in the animal models. There is no human with the lack of GIP3, but you can find people with mutation in GIP3. They survive and have this neurodevelopmental disorder, and they have affected different part of the brain.
Yeah. So tell me a little bit about like the different part of the brain and how it's affected by this
The GIP3,
mutation. So
yeah.
you mentioned that there's different mobility issues, but also autism. But to me, we have all these different types of neurons and different areas of the brain that do a lot of different things. Is Is there one particular type of neuron that gets affected from your research, or is it kind of just the whole system goes haywire?
Yeah, so from the human, what you see, it is a global affect all the brain. And basically, there are some part of the brain they are, we can say, they are more affected. There is a part of the brain called the corpus callosum, which acts on the cross both and connect both hemisphere of the brain, and that's thing very affected in this patient. Have very thin corpus callosum, and the cerebellum, which is control the motor coordination and is located in the back of in the base of your skull. That is also very reduced. The white matter, which in the brain is very reduced and the brain have different forms, no? So it is seemed it is global affected, this mutation, but in our animal model also we see the same. We don't see any particular neuron affected more or less. So we are very intrigued because what is affecting, this seems to be affected all the neurons similarly.
Okay.
Yeah.
So I'm really interested, how did you overcome this lethal mutation in the animals in order to actually generate this model?
Yes, this mutation, one that I'm studying, is not lethal in human, but if you remove
Okay.
total GIP3,
Yeah.
that is lethal. But this mutation it doesn't affect the GIP3 levels. They still remain in the brain, so it's not like when you remove all GIP3, but still the animal can survive. And if you do some cultures with the neurons are okay, and that allow us to do even behavior with the animal, and then
Mm.
we can see we can try to see if our animal model replicates some of the features of the human patient.
And
So
so
we're
far, have you been able to see a similar behavioral phenotype or behavioral look to how these animals actually behave in comparison to people?
Yeah, we are trying to work on that and analyzing the data of the behavior. We are finding some interstate data. So probably soon we will release a preprint with the data of the first characterization of this animal as a neuron as mouse model of this disorder. No, which is forgot to tell that these rare neurodevelopmental disorder have a name. No, it's a very long name, but it's called, like, the abbreviation is a NEDVA, NEDBA. So it's n- neurodevelopmental disorder with or without brain abnormalities. So
That is a mouthful.
yeah, yeah. But in short, you could call it NEDBA. So because there are several mutations affecting GIP3, we study only one, which is the most
Mhm.
common. Uh, but there are several mutations in this protein. So all of them is called NEDBA. Yeah.
So, when we're thinking about these mutations, I mean, it sounds like it's a very specific mutation, a very specific patient population. But even though these other ones are rare diseases, when I think of a neurodevelopmental disorder that involves the lysosome, I think of things like Hunter or Hurler syndrome.
Mhm.
And, it's, related to lysosome degradation, dysfunction, and that kind of stuff. But it sounds like this mutation leads to something totally unrelated. And I really want to kind of unpack this idea of what exactly is this protein mutation doing to the lysosome to make the neurons. You said they're fine if you
Yeah.
culture them. They're clearly not fine, though, if they're not working properly.
Yeah, yeah. So the previous research, when they start to describe this mutation, one of them, they did some preliminary tests where they put this mutation in zebrafish, and in the
Okay.
zebrafish, they realized that it's creating some kind of swelling in the neurons in the zebrafish, and they were accumulating some organelles like lysosome.
Mm-hmm.
And some other study, they took this mutation and they put it in C. elegans in the worms. And in
Hmm.
C. elegans in worm, also they're seeing they are affecting somehow the transport of this lysosome, because GIP3
Hmm.
basically is a protein. The lysosome need to move inside of the neuron to kind of, let's say, to to clear and help for the signaling, and they mature in the axon and they move to the cell body of the neuron, and then they do the job. No, so if this transport is affected, the clearance and the signaling inside of the neuron is affected. So that is the neurons start to look unhealthy.
So
So
to me, it sounds like this protein is super important. Like if we were to imagine that the axon of a neuron is like a road and it's
yeah,
a lysosome to continue this idea of it kind of being the garbage truck, even though we know it does more than that,
yeah,
but
yeah,
still, if, it's traveling down this road, this protein seems like it's really important for keeping it on the road. And it's almost like there's a flat tire or there's, a car crash or something like that. And then the lysosomes all get stuck
yeah,
in
yeah,
that area of the road, and then all the trash doesn't get picked up
yeah.
from the rest of the street.
Yeah, yeah. So yeah, that is a
Perfect.
good analogy. Yeah, you need to keep the highway
Yeah.
clean, and then you need something like to remove all the stuff and keep flowing, the information or the cars passing in the highway. Yeah.
So that makes perfect sense. That makes perfect sense for why this mutation would lead to those types of behavioral differences or, white matter differences if
Yeah.
we're not clearing garbage out of our neurons like we're supposed to. And I feel like that ties so clearly into what the rest of the Ferguson lab is also working on in terms of like Parkinson's or other types of neurodegenerative diseases.
Yeah, yeah. So that is in the so all the lab work with the lysosome. So I am particular focus with this protein. Um, and also since Sean already had experience with this protein before,
Hmm.
so we are trying to figure out now the mechanism, what is actually this mutation doing. No? And luckily we have also collaboration with the Wolverine Foundation. The Wolverine Foundation is a foundation here in the United States. Connect patient because it's a rare disease. There, beside these two papers, and there are like four papers now showing the clinical data with patients. The Wolverine Foundation is trying to bring people together and bring scientists to try to connect and see what we can do and help to uncover what are the mechanisms behind of this mutation in GIP3 particularly. So um, so they helped our lab and trying to study this disorder in GIP3. Recently, there was, I think, it's the first paper with a cohort from United States with patient in United States um, from a lab in Harvard. Wendy Chun is the head of that lab, and she's an amazing um, neurodevelopmental disorder researcher. And you can see that the patients are very similar to the one previously described.
That's
So
awesome. I mean, not awesome, I suppose, that we're finding patients that have this disorder. But for
yeah,
anyone who's watching and listening, I feel like if you're not that tapped into rare disease research, it's actually really hard sometimes to get funding.
yeah,
And, these poor patients, these poor families, because there's just so few people who have these genetic diseases or genetic disorders. They're hard to study. It's hard to find patients to
yeah,
study. And then eventually, you hope you're going to have like a cure or a treatment or a gene therapy or something. But then it's even harder to test that potential new treatment because there's nobody to test it on. And so I know like families are obviously like really desperate for research and help. And so it's awesome to know that there is this foundation and that you guys are connected to be able to help with that research for these families.
yeah. And one of the good thing of this collaboration that we can really look and do zoom into the cells of the patient, because we work with the stem cells and we can reprogram
Mm.
the stem cell from the patient into the cell of interest. Now in my case neurons, and we can really look into cells of the patients. No? So we can now correlate what we see in the animal models, in cell lines and the neurons from the mice model, and see if that is similar to what we see in the patient cells. So
So I feel like
it-
that really like leads into like my next question for you. you've done research in a ton of models now. Like you've done research in rat, you have a lot of experience in mice now. You're talking about induced pluripotent stem cells being developed into neurons in vitro in order to study these different diseases. Can you really tell us, like, what are the benefits and drawbacks of maybe using a mouse over a patient induced pluripotent stem cell line? Like, what can we get from a mouse that maybe we can't get from a patient stem cell line versus what can we get from a patient line that we can't get from the mouse?
Yeah, so the mouse model is interesting because we have the whole organism and we can look into different type of neurons of all body and look very into details and perform some techniques that will be very difficult to do with humans because we can observe the development of the mice when they are young, middle age, old, and take a sample from the brain or from different regions of the nervous system, analyze the behavior, different type of behavior with exhausted behavior, which is with the patient is will be very difficult to get. No? And now, once you have this data, and then you have like an idea of what could be the molecular mechanism. You go to the iPSC or the stem cells from the patient, reprogram this into neuron, and they say, "Oh, what I found in the mouse kind of replicate what I see in the human cells," and then you can start to elaborate a better model for the disease. No? And and again, yeah, like you mentioned, treatment and drugs, because that is one of the caveats of biomedicine. And sometimes the things that works in mice on cells on mice, cells that derive from mice don't work in humans. So you need like a human model to try if
Hmm.
you want to find the molecular mechanism and you try to find some new drug or new therapy. So it's very valuable. I think nowadays, most of the labs they working in disorders, they have both. No, yeah,
And so, with your induced pluripotent stem cell lines, I have a couple questions. I'm a
yeah,
stem
yeah,
cell person, so I mean,
yeah,
I could ask you. We could talk about stem cells for a whole other
yeah.
episode, but I'm so intrigued. You know, I honestly don't know what the role of the lysosome would be in reprogramming, but I know that it can be really challenging if you have like a mitochondrial DNA mutation in order to actually undergo reprogramming. And I just have like a real curiosity question. How hard is it to reprogram these cells from these patients because of this mutation? Is it like totally normal? For anybody who doesn't know, reprogramming itself
Yeah.
is not the most efficient process. So anything that makes it more challenging is usually not fun. So I'm so curious.
Yeah, not so we noted this mutation, at least the one that works, affect neurons. So when the cells are in the pluripotent state, when they are in the stem cell, they don't have much G3. The
Okay.
G3 start to express and increase the level when the stem cell you differentiate into neurons. So there is no big problem to have a stem cell with the mutation. No? And now there is several new tools for studying stem cells and reprogramming to neurons that is very easy now. There is called the I3 neurons. They were developed by a group in the NIH. Basically, you just add a drug and wait several days, change of media, and then you have neurons. Like in 10 days, you have like
I
a
love that.
neurons. So before, like you mentioned, I know what you were saying. Before it was super tricky because you needed to put into the stem cell several factors and then wait for 43 days to have neurons or other type of cell. But now, most of the group that study neurons, they can use that. But there are other people that they use the previous version now. When you
Yeah.
put factors and you wait
Yeah.
longer time, but there is a way to have neurons in 10 days. So very fast.
Easy.
Yeah, easy.
I
Yeah,
My
yeah.
second question for you is, have you taken any, you mentioned developing these cells into neurons specifically, but we have like all these other different types of cells in the brain, and you mentioned earlier that it's not just the neurons that are having problems often when it comes to this type of disease. So, have you guys done any brain organoid research to look at a bunch of different cell types all at once or anything like
Um,
that?
yeah, no, No, I would love because the organoids to work with organoids is a little bit more complicated, no? Because you need,
Hmm.
and also you need more days of
Yep.
incubation. You need to wait for the organoid to really show you some kind of resemble of one part of the brain that you are interested.
Yep.
And that is so only speaking about the organoids with neurons, no? If you want to put the complexity of microglia, astrocyte,
Uh-huh.
and do
Extra.
co-culture, yeah, extra, it is, I think, a little bit more complicated. And you need the special system machines
Hmm.
inside.
Hmm.
So, yeah, increase a little bit the level of complexity. But for the question that we have, or I have in my project, I think we have the tools to work. And so far, the animal model has been great. And with the stem cells that we have, and we can differentiate into microglia, macrophages, so immune system cells
Hmm.
and neurons. So with the tools, we are okay. But would it be nice to work with organoids? But it takes so much time and
I
also
know.
money.
Yeah. Time,
Yeah,
money,
and money. Yeah, yeah,
very specific types of expertise.
yeah. Indeed. Yeah,
And it's not, yeah, it's not just, I can differentiate or I can develop cells to a particular cell type. It's, how
yeah,
big are they? What do they look like? Like so many extra steps. And do
yeah.
I
And you
want to
need.
put them in a scaffold or like a hydrogel or something like that?
Oh,
Like
yeah,
so
yeah,
many different questions that come
yeah,
up. I was just so curious if maybe that was like a potential avenue that you guys have thought of. But
yeah,
I mean, I think that's a really good opportunity to also mention that, like, just because there are really fancy tools doesn't necessarily mean that that is the best or most effective tool to answer the research question.
yeah,
And for those of us who might be watching or listening who aren't scientists, like sometimes we need to really not just think about the money question, but really the what is the question that we're asking, and do I need it to be so fancy when if we're looking for a really specific mechanism inside of a neuron and about like what is the lysosome doing, what's happening, I probably just need to look at neurons. I maybe don't need
yeah,
to look at anything else until I figure that out.
yeah. And then if you can add that, we are one is one type of cell, but then you look, okay, one signal inside of the neuron. Okay, then I'm gonna look only one organelle,
Uh-huh.
and that is already complex to try to solve a problem. Imagine you put a level of complexity up, no? So
Yeah,
depending on your question, you use the tool that you have available, and sometimes like a simple question is how the lysosome are affected by this mutation with these two models that we have is enough, I would say.
definitely. I
But of course,
so
the more the
agree.
merrier, no? If we will have infinite amount of money, okay, yeah, we can use all the models available.
Yeah, right.
Yeah,
Use
yeah,
them all.
yeah,
I
yeah,
mean, maybe one day, maybe someone will also pick up this research or you guys will collaborate with somebody who already has a really great organoid system. And then you
yeah,
guys can just like fire the pluripotent stem cells into their system and see what happens. And then just like wash your hands of it and be like, report back.
yeah, yeah, exactly. Yeah, no, that would be great. Yeah,
That would be awesome.
yeah,
But my follow up question is, you've talked about behavioral studies now in rats as well
yeah,
as what you kind of work on now more in mice. And I'm really curious, how exactly do you study a mouse or a rat's memory? Or how do you study a mouse's ability to, I guess, show an autism phenotype? Or like how do you do that? They're a
yeah.
rodent.
Yeah, so I love to work with rats. They're very cute, and then they behave better than mice. But basically, you do different tests depending of the lab. You will have different flavor of the memory tests the labs do. The most classical is called the water maze, which is a pool. They have two platforms, and you have to train the animal to find one hidden platform, and the animals they have memory problem, they take more time to reach to the hidden platform because the animal once that you train it, they know the way to find that platform. So and they look cues around the pool to see where it is, and then they
Hmm.
are very smart. Rats and mice are very smart, and that is the classical um, memory test that is very used in Alzheimer's disease, an Alzheimer's model. And the other typical is when you train your animal to recognize two objects and then you remove one object and you put a new one, and the animal, since already know the other, will spend a lot of time in the other new object sniffing and looking around because the other, that is also classical. And it's called the Y maze, which is a test in the shape of a Y, and you cover one, like a little corridor, and you let the rat explore or the mice. It can be used for both, like a 5 minute, and the animal will explore because they look like they like the novelty. And then you take the mice or the rat and wait, like depending on the time, 2 or 4 hours, and then you put it back. But now you release the block corridor, and then the animal will reach the Y and then say, "Oh, I already know the left, so I'm gonna spend a lot of time in the right. What was in that side I didn't see before?" No, and then you make like a ratio how many times they enter one arm of the test. There are plenty, like there are different models, but
No,
those are
yeah.
the easy to do and not much time consuming. Now there are even fancy ways to test memory and with the screen and all stuff.
Oh,
And
wow.
yeah. But
I mean, it sounds like the rodents are maybe smarter than we give them credit for.
yeah, there are a very nice paper. The people should look. The people that train rats to drive little cars. The rats
Oh, I
drive.
remember that.
Yeah, no, the rats are amazing. Yeah,
Right?
so
I mean, I don't know who justified that in a grant, but thank you for the
yeah.
rats driving cars
Yeah,
grant
that
money.
that was great.
Oh my gosh.
Yeah.
I love that. So, thinking about your future, you've been an academic for, over a decade now, and now you're working on rare diseases, neurodevelopment, iPSCs, mice, all these different things. When thinking about maybe opening up your own lab one day, is that what you see yourself doing in the future? Do you think you're going to want to still work on the same problems that you're working on now, or do you kind of see yourself, no, where are you heading?
Yeah, I would like to have my own lab, and I've seen recently there is a lot of work in lysosome in neurodegenerative disease. But I always think about there are many things to discover about lysosome during neurodevelopment and the neurodevelopmental disorder associated with some of the proteins that control
Hmm.
lysosome, either outside or inside of the lysosome and how they move. So there are rare disease, and also, yeah, I think there are not many funding, but the rare disease, we have many rare disease that impact the lysosome all around the world. The one that you mentioned today, and there are different mechanisms affecting.
Hmm.
So I would like to have a lab that um, study lysosome in neurodevelopmental disorder that can cause autism or intellectual disability. So, and having the experience of rats and mice, and now with the iPSC, I think I can have a set of tools that allow me to have answers in this rare disease.
I mean, I definitely see it. And I feel
Yeah.
like, you're obviously the expert on lysosomes and neurodevelopmental disease, but it feels like even what you mentioned about not only is it the garbage truck, but it also signals to different organelles inside the cell. Like that feels like there are so many new avenues and roles that the lysosome might play during development that maybe we just don't even know about yet.
Yeah, and also, some people now even are wondering if some neurodegenerative disease, they manifest later by aging, but they are always inside us. Those, problem. It's just an aging problem.
Hmm.
Uh, but we never, since there are aging problem, we never look into the development, no? So we don't know how that's affected. Probably we don't manifest those problems, but
Yeah.
or maybe,
Or maybe they do
manifest in a way.
manifest. We just,
Yeah,
yeah, we just don't know how they do.
yeah, exactly. So I always wondering about that, like those mutation, some genetic mutation that cause neurodegenerative disorder, they manifest when the aging, uh, but what about between our 20 and 30s or
Yeah.
even younger? And so
No,
I am
definitely.
curious to to explore more about that topic.
That sounds awesome.
Yeah,
So, before we wrap, I always think it's super important to not only talk about the science, and we've spent a lot of time talking about science, but it's also, I think, really important to highlight that scientists are just regular people. We just happen to have a really cool job, I think.
yeah.
So, Pablo, can you tell us, what else other than the science gets you excited to get up in the morning? Do you have any like really fun hobbies? Like, what makes you you when you're not in the lab?
Yeah, few years ago, like 3, 4 years ago, I started to get into running, and
Nice.
yeah, and so then I start to compete just in 5Ks, And then I say, okay, I'm gonna go for long distance. So, and I already ran 2 marathons and like a 5 half
Wow.
marathon. I'm preparing now for the Philadelphia Marathon in November. So that keep me like, training, doing. It's exhausted too, but it release it's kind of other type of of you know um mental work. At least you
Yeah, I was
enjoy
gonna
it
say,
at the end.
right? I like to think as well, like running, not to, bring it back to the science,
Uh,
but it's like really meditative. So I would imagine like
yeah.
you're probably your brain's probably working on problems while you're like focusing on the running, but like it's working out the science problems that you need to tackle the next day or the next week or whatever.
Yeah, at the first 5 kilometers are always painful, so you don't think
Mhm.
anything about like, oh, this
So
is
true.
painful. I don't know why I'm doing it. And then you
Yeah.
enter to the like a flow, and you run. It's like I am okay, and then your mind, just think anything. And then at some point, or to me, it's like I don't think in anything. It's like it's just meditating. When you pass like
Yeah.
the 12 kilometer, 15 kilometers, you are like listening music, and there is nothing in your mind. So that is very relaxing. So I do that, and then I play guitar in my free time just to, songs for me that I like to play just to
Mhm.
relax stuff. Yeah. And hang out with my friends. But yeah, the running keep me busy a lot.
No, that's amazing.
Yeah, yeah, yeah.
No, I like I said, I think I am a big believer in the fitness to help de-stress and all that kind of stuff. And
Yeah, yeah.
I think there's a lot of scientists that feel similarly.
Yeah, yeah.
So with that, I just want to thank you so much, Pablo, for being on Science with Spice Between Experiments. It's been so cool to talk to you about lysosomes, neurodegeneration, neurodevelopment, things that I really honestly knew nothing about. So thank you so much.
Yeah, thank you, Daniel, for the invitation. And yeah, I will keep seeing your reel and repost it to other people.
I
I think you
appreciate it.
communicate very well with science for all complex, topics, and you make it look very easy. So I think, yeah,
I really
yeah,
appreciate that. Thank you.
yeah,
I want to thank Pablo so much for coming on and talking to us about all things lysosome and neurodevelopment and really shining light on how important it is for us to really dive into rare disease research and how important lysosomes are for our developing brain. If you want to get more episodes of Science with Spice Between Experiments, you can check them out on YouTube or anywhere you get your podcasts. You can also find me, Dr. Spice, your host, on Instagram and on TikTok. Until next time.